So for chapter 11, we're going to be looking at membrane structure. So we want to keep in mind that cell membranes act as selective barriers. So they're known as being selectively permeable. In this figure with the panel A and B, we can see that we have a bacterial cell in panel A and a eukaryotic cell in panel B. And so with a bacterial cell, which is prokaryotic, you're going to only have the plasma membrane. With a eukaryotic cell, they're going to have the plasma membrane, but also internal membranes. And so we do want to keep in mind that when we're talking about cell membranes, we're talking about the membranes within the cell, which can include both internal membranes and the plasma membrane. So if we're talking about proteins, or if we're looking at the plasma membrane, we want to make sure that we're using the correct terminology, that we're not calling, yes, it is a cell membrane, but if it's something specific to the plasma membrane, that we're using the proper terminology. So when it comes to assessment pieces like the exam, yes, you will lose points if you're talking about a transport protein that's on the plasma membrane and you just say cell membrane and it's something it would only be on plasma membrane. Okay, so do make sure that if you're talking about the surface membrane, that we're talking about the plasma membrane. Right. So when we look at the eukaryotic cell, there's lots of different organelles that do have membranes, like the endoplasmic reticulum. We also have vesicles like lysosomes, perisomes, transport vesicles. There's also the mitochondria, Golgi apparatus, and nucleus, right? So all of these are membrane bound, and this is lacking in prokaryotic cells. So prokaryotic cells aren't going to have those internal membranes. They are still able to synthesize proteins. They still have DNA. They still transport molecules, but they don't have the vesicles and these organelles to do that. So the plasma membrane, that cell membrane that's on the surface of the cell that distinguishes the outside environment from the inside environment, it has a number of cellular functions. So the first one would be receiving information. So this can be done through protein receptors that are on the surface, but there would be something going on the outside of the cell, and that information needs to be taken in by the cell so they're aware of what's going on in their environment. And again, receptor proteins are a common way that this is done. The plasma membrane also serves as a way to import or export small molecules. So if we're importing that could be done through processes like phagocytosis, pinocytosis. These are all endocytosis. So endocytosis is the general term where we bring material into the cell. And so this is where some of that plasma membrane can blub in to the cell and take some of the material from the outside environment in. Okay, so vesicles would be formed. So again, endocytosis is the general term. Pinocytosis is when you just have a liquid coming into the cell. Phagocytosis is a term specific to white blood cells or leukocytes or immune cells, right? So immune cells undergo phagocytosis when they internalize bacteria or foreign material. Okay. For the export of small molecules, this is where something has been synthesized within the cell, or is in excess, or is a toxin, and so it's put into a vesicle, a transport vesicle, and is exported out. This would be known as exocytosis. Okay, so both endocytosis and exocytosis is where what we use for some of the though they're small molecules, larger molecules, we can also have simple diffusion or active transport using transport molecules also. So vesicle movement of material is not the only way we can have it so that it's channel pore specific. So this is where proteins would be utilized, or we could have simple diffusion for small ions through. And then lastly, another function of the plasma membrane is to be able to move and expand. So if we think of like an amoeba, they're able to rearrange their cytoskeleton in order for that movement to happen, but they're going to have anchor proteins that are in the plasma membrane that's going to allow for that movement. And we'll look at that later on when we look at the different types of proteins that are on the plasma membrane. And one of those will be anchors, and we'll see how that actually causes the cytoskeleton to be anchored to the plasma membrane. So when the cytoskeleton gets rearranged, the plasma membrane is also able to be dynamic and fluid-like and move with it. We also want to keep in mind that if we have cells dividing, that we're going to need to be able to have that flexibility, that movement, that expansion for the plasma membrane in order to support cell division, because we have this increased amount of material, but then that has to be divided between two cells. Okay, so when we look at the plasma membrane, that is going to be made up of what's known as a lipid bilayer or a phospholipid bilayer. Within that, we also have protein molecules, and we'll talk more about these protein molecules later on, but they're going to help with the function, so like receptors, transport, proteins, channels, also anchoring proteins. Focusing more on the phospholipids, which we'll do in the first part of this lecture. If we look at the phospholipids, at the lipid molecules, you'll notice that they have a kind of circle red area that's the head of them, and that is going to be hydrophilic. Okay, so it's going to like water. So because these are positioned on the outside, because these layers are coming together and the tails are meeting and the heads are on the outside, because it's hydrophilic, it's going to be able to interact with that aqueous environment that's inside the cell and also outside the cell. The tails are hydrophobic, and because we create this bilayer, it then allows that hydrophobic area of the tails to be tucked in, so that they're not interacting with the aqueous. They have the head, the hydrophilic region, to buffer them and protect them against that water. So next, I'm going to skip over this slide that has a video. I will post the video under the content for Unit 1 with the lecture slides and files so you can view it. But I will talk briefly about it, so that this might be a good time to pause this video, the audio lecture, and then go watch this video, and then come back to hear my kind of chat about it. Okay, so hopefully you watched the video, and what you would have noticed is that as they applied pressure and they pulled on that plasma membrane, that the plasma membrane was very dynamic and flexible. It moved, and so we tend to think of plasma membranes and cells in general as these very static, non-moving structures, right? And the reality is that there is a huge amount of movement and response that's going on by the plasma membrane and the cell. So we see these figures in the textbook, or maybe we see images that are captured through microscopy where the cells have been fixed, or these are just, they're not active videos, they're just a snapshot of the cell's life. And so I think we a lot of times think of them as these kind of, that's what they look like from the drawing in the figure. But molecules are constantly moving and rearranging inside a cell. And that video shows an example of how that plasma membrane is rearranging and moving and responding to this stimuli, the person pulling on the plasma membrane. So all this rearrangement of the cytoskeleton is happening in order for that to occur. Now, if we have a cell that doesn't change its shape, and we're doing like live imaging, that could be because it has a cell wall. So prokaryotic cells like bacteria and also plants have a thick cell wall, and so that cell wall allows it to maintain its shape. But if we look at stressful situations like in a plant or in bacteria, we would see that a stressful situation being that someone forgets to water them, they respond, they change, right? So we have a wilting plant, and that would be because there's not enough water in there. But if we actually looked closely, we would see that the plasma membrane is actually responding, and that it's moving away from that cell wall. And if we were to then water the plant, that plasma membrane now, because of all the water in it, is going to respond to that internal pressure, and now it's pushing up against the cell wall. Now the plant is standing up, doesn't have that wilted appearance. Okay, so again, if we have a cell that's not that doesn't look like it's being dynamic, we want to say, why does it not look like it's being dynamic? Is it because it's been treated, fixed, so that it isn't dynamic anymore? Are we just looking at a snapshot? Or is there another structure that gives it this more rigid appearance, being the cell wall? So if we look at the basic molecular composition of the lipid bilayer, looking at these phospholipids, what we would notice is that on the polar head, the hydrophilic region, we're going to have three pieces that are coming together. So we have what's in the blue, the chlorine, is specific to phosphatidylchlorine, but there are different types of phospholipids. So again, that blue piece is going to change. So it's like a side group, right? Or known as a head group. We then have the phosphate and glycerol. So phosphate and glycerol would be found on most of the phospholipids. There's an exception, and we'll talk about that in the next figure. Next slide. And then we have our nonpolar tails, and so most of our phospholipids are going to have two tails. Again, there's always an exception. And one of the tails ends up having a cis double bond, and that cis double bond causes like a kink in one of the tails, and so you end up with one of the tails almost like it's kicking out. So you can see here in the space field model in panel C how the one tail almost looks like a leg, and so the phospholipid is like dancing. And again, that's because of the cis double bond. Okay, so this figure is just showing us the different phospholipids that are present in plasma membranes and also cell membranes. And so you'll notice that the majority of them do have two fatty acid tails or hydrocarbon tails. We then have the glycerol molecule, a phosphate, and then we have that head group that's unique to each of them. Okay. Obviously, there's exceptions to it. And I won't put on the exam the different ones and say identify it. You should just know that there is different phospholipids that are present, and we'll talk a little bit more about that after the next slide. So cholesterol is also an important component of phospholipid bilayers. So if you look at cholesterol, where we have that polar head group, we have this rigid sterile ring structure. So this doesn't allow for a lot of flexibility, which becomes important in a moment. And then we have a nonpolar hydrocarbon tail. Okay. And so what happens is that the cholesterol integrates in between the phospholipids, and it actually helps to stabilize them. So it actually makes the membrane more rigid than what it would be if there was no cholesterol present. So different organisms are going to have different amount of cholesterols. So they're also going to have different ratios of the phospholipids present in their membranes. But if we think about phospholipids, they're more like vegetable oil. okay, where cholesterol is a little bit more like butter. Okay, so this is just an analogy here. So if you have butter out in room temperature, it's a solid, okay? So it'd have to be hot in the sun to like melt or microwave it. Where phospholipids are like vegetable oil, and so if they're at room temperature, they're going to be liquid, okay? So by adding the cholesterol in, it actually makes it so the membranes are more stable. So if we were to take some of the vegetable oil and some of the butter and to homogenize and mix it together, we would have something that was more consistent with our plasma membranes. And that's kind of like if you had like margarine. So margarine, I can't believe it's not butter. Actually has butter in it. So that's kind of like a mix of oils and butter. So that it's softer than butter, but it's still at room temperature would be solid. And so again, keep in mind that our plasma membranes, our membranes are very dynamic and fluid and they can move, but we also don't want them to be liquid at room temperature, right? So the cholesterol helps to stabilize that and prevent that. We also want to keep in mind that we also have proteins that are embedded in, and so those proteins are also going to kind of dilute out that more liquid-like characteristics of the phospholipids. So what this slide is looking at is the amount of energy that's in order to create a lipid bilayer. So keep in mind that inside the cell, it's going to be majority water, but we're going to have other components inside the cell. Those components may be hydrophobic, just like the phospholipids have the hydrophobic region. So if we are to put a molecule like propane here that is hydrophobic in water, it actually naturally starts to kind of force those water molecules away, kind of creating like a droplet around it. And so if we think about if we were to take membranes and put them into water, they're naturally going to want to create an environment in which the fewest contacts with the water is made. And this actually creates a low energy, minimal energy in order for that to happen. Okay. And just like we saw, proteins are going to fold using the least amount of energy. Same thing with our membranes. Those need to be formed with the least amount of energy. We want to save energy for other things like catalyzing reaction, creating those peptide bonds, not unfolding and such. So this figure looks more of it kind of stepping back at those phospholipids. So if we were to look at the lipid, if it only had one tail on it, the natural tendency would be actually to create these little meniscus droplets being formed, okay? But because there's these two hydrophobic tails, and you need to have space to put those two tails in, plus we have a little kink there because of that cis double bond that causes that one hydrocarbon tail to kind of kick out, you're not able to create that meniscus, that droplet. So the natural tendency is to actually form a bilayer. So to turn two layers of hydrophobics in that are protected by that hydrophilic head. And because we need to have edges, edges would be areas that would interact with the water. The natural tendency is to actually go and turn that in on itself. So as we see on the panel on the right, we have this sheet. All those edges on the sheet potentially would be interacting with water, and so it's going to want to turn in on itself so that we end up forming a cell, a sphere. so that there are no areas, no edges that are exposed to the water. So this is very energy favorable. So this is a low energy arrangement. So if we had regions in our cells that were exposed to water, we would want to, the natural tendency is actually just to repair, to fill it in, to make a fluid movement where sealing of that damaged area would be removed, right? And so we see that in processes like endocytosis and exocytosis, so that there is no exposed edge of the plasma membrane. Right, so the scenario I was just talking about is if we have this tear in the bilayer, well, why don't we just take those and turn them in on each other to create this hemimeniscus cap, right? So we said if there was one hydrocarbon tail, like in detergents, they can form a meniscus, they can form that spear. But there becomes an issue when you have two hydrocarbons, because then now there's going to be this gap between that hydrophilic head, and so that's areas where water could be exposed, right? So this, though it might protect some of that membrane, it's not going to protect the edges against exposure to the water, and that's not what these hydrocarbon tails like. So they would try to rearrange themselves in some way. So that's not a favorable environment for them to exist. So again, what I mentioned is there's this resealing movement of the plasma membrane in order to seal off tears in that bilayer. And again, we see this if you were to study the process of exocytosis and endocytosis, this repair process, there's proteins called snares that are involved in this process so that this bilayer doesn't get exposed and repairs itself. So liposomes are an example of an organelle, a structure within inside the cell that is a way in order for us to look at and study lipid bilayer behavior. So liposomes are really just these membrane structures that are made up of phospholipids. That's liposome, and they're being exposed to water on the outside and inside. But because we have this bilayer, none of that hydrophilic region or hydrophobic region of the bilayer is actually in contact or exposed to the water. So again, we can study different properties of lipid bilayers by creating liposomes and then study them. So we can add in proteins, we could add in different components, we can see how large they're going to grow to, so we could try to make larger liposomes or smaller liposomes. So there's researchers who study lipid bilayer behavior. There's also computer modeling that can predict how fast repair can happen. So lateral diffusion is that moving of these phospholipids. Again, it can be done because we need to move them to different areas because maybe there's been some kind of damage done, but just in replacing. So just like any organelle, any structure, any protein in your cells, they are going to wear out and they're going to have to be replaced. Okay. If we're where we're responding to something, there may be flexion that's needed between those hydrocarbon tails to allow more flexibility within the plasma membrane, or some kind of rotation that occurs. The flipping of a phospholipid from one layer to the other layer rarely occurs, and we'll talk more about that later. So here we can see the lipid composition for different cell membranes. So, you know, if we look at a bacteria, which is in this last column, E. coli, versus our, human cells, there's going to be different. It seems pretty obvious that there would be differences between a bacteria and us, right? But even within our own cells, there's going to be differences. So you can see the chemical composition between a liver cell and a red blood cell are going to be different when we look at the plasma membrane. In myelin, when we look at structures inside a cell, the mitochondria versus endoplasmic reticulum, there's also going to be differences. There's going to be differences between them, between those different organelles, and also the plasma membrane of the cell. Okay. So when we add cholesterol in, right, we're going to end up stabilizing the plasma membrane. But also, what happens is that we get what's known as immiscible phases. So if you look on panel A, we can see how the dye that's been added is spread throughout the membrane. When we add the cholesterol, what we start to do is get these clumps or rafts. So these are known as lipid rafts, so that we get these areas where these phospholipids are going to kind of group together. And so in the next couple slides, we're going to talk about lipid rafts. Okay, and again, this work was done in liposomes because they're going to be easier to study because they're much more simplified than looking at a whole entire cell. And so here we can see how we have these rafts that are being formed. Okay. So what ends up happening in these lipid rafts is that we can find that there's proteins, usually cell signaling proteins, that are found there, right? So we might have our proteins that allow for transport inside the cells, or our ions or channels, but signaling proteins tend to be found on these lipid rafts, and it's a way to actually have all of those proteins, these molecules that are needed for these signal transduction pathways, or these cell signaling pathways, to be found in like one area. Okay, and so we don't want these to be evenly distributed over the whole entire surface of the cell, because what's the chance that, if we're looking at a cell signaling pathway, there might be, 3 or 5 or 6 different proteins, kinases, receptors that we all need in one location. And so if these were all evenly spread throughout the surface of the cell, the likelihood that these proteins that are needed for a cell signaling pathway would not be found in one location, right? And so by creating these lipid rafts, it concentrates those molecules in the area, right? So if we think back to the figure that we just saw, in those red areas where we have these lipid rafts, that's where we're going to put our signaling molecules. In the other areas, that's the areas that didn't have the right staining, that's where we can have our transport proteins, right? So it allows for cells that are proteins that need to be together. If they stay on the lipid raft, they're going to be together. Okay. Another characteristic of the lipid bilayer is that it's asymmetric. Okay. So if in this figure, we have the color coding of the different phospholipids. And if we look at these different phospholipids, we'll notice that the red and brown phospholipids are going to tend to be on the extracellular surface, where our yellow and green phospholipids tend to be on the cytosol side, or the intracellular side. You'll notice that the phospholipids that are inside the cell are the ones that tend to have a charge to them. And this is important because some of these cell signaling molecules that are intercellular need to bind to a negative charge. So protein kinase C, or PKC, would be an example that needs to be able to bind to these negatively charged heads. So how does this pattern, this asymmetric distribution, occur? So when lipids are synthesized, they are more or less random in their distribution. Okay, so there's actually an enzyme scramblase. Scramblase, you can see that down towards the bottom of panel A, actually transfers in random phospholipids from one layer to another. So in your ER, you're going to end up having a mosaic of phospholipids. By the time the membranes have moved into the Golgi apparatus, this is where flippase, these two enzymes, scramblase and flippase, make me chuckle. So flippase actually will cause there to be a transferring of phospholipids so that we get the difference, so that what would end up being extracellular is going to have a different composition than what is going to be intercellular or on the cytosol side. Okay. And I would like just to add that with this figure, I do think the visual of this is a little difficult because you have these arrows kind of moving in the opposite direction of what you would think of. So if I was writing this textbook, I would probably change this figure so you were getting more of a visual of inside an actual cell. Okay. So if you have issues with this figure, please let me know because I'm more than happy to talk about this in class. So one of the things we want to keep in mind is that as things are moving through the cell, some proteins are going to be marked and labeled as having a final destination. Where are they supposed to end up going? And so they need to be put into the right location in the ER, in the Golgi, in the transport vesicle to end up in the right spot or orientation. Okay, so what we're seeing here is that what is inside of a transport vesicle, or what's inside the lumen of the Golgi, is going to end up being in the extracellular fluid on the outside of the cell. So this figure has the inside, the lumen of the Golgi marked in the red, right? And that ends up becoming the non-cytosolic face or the extracellular face of the cell membrane, or the plasma membrane, where the cytosolic face is going to be the green layer, right? So for the lumen, for our transport vesicle, and then also for our plasma membrane. So that if we had a membrane protein and we have this blue head on it, that it needs to be in the lumen of the Golgi, the lumen of the transport vesicle, in order to get to the surface of the plasma membrane, with that blue head still being on the outside of the cell. Okay, so that orientation, the topographic, the shape of the membrane needs to be preserved in that vesicle transport so that the orientation is right, because we're not going to have that ability to flip once we're out on the surface. Okay, so the flipping is going to occur in the Golgi of those membranes, the arrangement of it. So we can also have glycolipids that are found on the outer surface of plasma membranes. So these are going to have sugar structures on on the heads. Okay, so a little bit different than the phospholipids. Glycolipids are going to have sugars that are present are carbohydrates. We can also have proteins that on their extracellular domains, they get glycosylated, so they're going to have sugar residues. So what this ends up creating is a carbohydrate layer or a glycocalyx. Okay, so glycocalyx is a term that you should know. Really important and looked at in microbiology with bacteria, but your human cells also have glycocalyxes, and so the sugar layer is really important. One, it's a way in which bacteria can actually hide from your immune system, because if there's a heavy amount of glycocalyx, your glycocalyx and my glycocalyx and a bacteria glycocalyx looks very similar, right? And so it makes it tougher for the immune system to actually recognize the pathogen, the bacteria, versus like your own cells. The glycocalyx, these sugars also can be used for nutrients, so especially important in bacteria. So if bacteria do not have a food source because maybe they're living outside of a host at the time, they can actually utilize the carbohydrates that are in the glycocalyx as a food source. Okay. So again, some phospholipids are actually utilized in cell signaling. So we had talked about that in previous slides. We're going to have that negatively charged phospholipid, and that negatively charged acts as a way to hold a phosphate. So you can see in this figure how we have this phosphorylated phospholipid. And so when we're looking at cell signaling, again, this would be present like on a lipid raft. We're going to have this external signaling, and through phosphorylation events and a kind of cascading of activation of signaling proteins, we end up relaying this information that was received from the outside of the cell into the cell, so that then we can have a response by the cell. Okay, and we're going to look at this later on the course. We'll look at the cell signaling in one of the later units. So looking at some of the different proteins that are embedded in the plasma membrane, I've been mentioning them throughout the lecture, but just to kind of formally put them, on one page, we can have transporters or channels that will allow material in and out, right? So one of the functions of the plasma membrane is to have to allow to import and export material, and so this can be accomplished through transporters and channels, also through vesicles, and also through diffusion. Another type of protein are anchors. So here we're going to talk a little bit more about them in a couple slides, but here we can see these anchors holding different fibers to the plasma membrane. So this allows that if these fibers are contracted or rearranged so that the fibers become shorter or longer, this would cause changes to the plasma membrane. And then we have receptors. So receptors, we're going to get a signal from outside the cell, extracellular, and then the receptor then causes changes to kinases or other proteins, other molecules, sometimes through phosphorylation, so that we end up getting that cell signaling cascade pathway. And then lastly, we can have enzymes on the surface of the cell. So sometimes these enzymes will catalyze reactions. So when we're looking at like viruses, a lot of times their receptors are coupled with enzymes, and those enzymes then aid in allowing for the virus to enter into a cell. Okay. So one portion of the enzyme might be sensing like a cofactor outside of the cell, and then inside it's going to take substrate and make product is also another example. So this figure just looks at some of the different plasma membrane proteins and their functions. So I would say these ones are fair game when it comes to the exam. We'll talk a little bit about integrins in a little bit, but we'll also be talking about some of these later on in the semester. So when we cover the different chapters. So how do proteins associate with the plasma membrane? So we can have these transmembrane proteins. So here we can see how we have the alpha helixes. So those alpha helixes will make for great transmembrane proteins. But we can also have what's known as a beta barrel. So the last, in panel A, that last image you're seeing there under transmembrane, you can see those beta sheets. So this is an arrangement of beta sheets that actually create a pore. And again, we'll talk about that in just a little bit. We can also have a monolayer association. And so this is where only the protein is only embedded in one layer, so the alpha helix can get embedded into the one layer of the plasma membrane. We can also have lipid linked, right? So if we have lipids, we can create bonds between that head group, and that would then be linked. So we just want to keep in mind that if we have an enzyme that can cut lipids, that protein now is no longer going to be attached or anchored onto the plasma membrane. We can also have proteins that are attached to other proteins. So again, this is really important in cell signaling. So we can have a transmembrane protein that will then bind to another protein, either intracellular or extracellular. So if we keep in mind the information we learned in Chapter 4 about proteins and how they end up folding, remember that if we look at the alpha helix, these can have, for one that is going to be a transmembrane protein, the alpha helix would have the hydrophobic amino acid side chains on the side, right? And so then there becomes a natural tendency for that alpha helix to be embedded in a membrane so that it's not interacting with the cytosol or the extracellular space. Okay. The transmembrane alpha helixes tend to be about 20 to 30 amino acids long because it has to be able to, it runs the length of that bilayer. Okay. In some cases, these alpha helixes can create pores. So you can see how we have 5 different alpha helixes. And so this is going to create a pore, a hole in the middle that would allow water molecules to go through. So for this alpha helix, one side would probably be hydrophobic and the other side would be hydrophilic, right? So we can begin to predict where transmembrane domains might be present, where we might get these alpha helixes by doing hydropathic plots. So what they do is they look at the peptide and its ability to move from a nonpolar solvent to water, and how much energy is going to be needed in order for that to happen. And so if we end up with a positive, which is shown in the darker green, that would indicate that that region would be hydrophobic. Okay, and so we would predict that that would be a transmembrane domain. So we can see that bacterial rhodopsin has a number of these transmembrane domains that are predicted. Okay. So we also want to keep in mind that transmembrane domains, transmembrane proteins are not just alpha helixes, that we can have beta sheets formed and that they would create pores. And so again, here we can see this beta sheet anti-parallel. And because this is an extensive beta sheet throughout the protein, when it comes together, it ends up creating this pore. Okay, the size of the pore would then indicate what might be able to go through that. Okay, but again, the hydrophobic regions would be where the phospholipid bilayer would protect it from any of the aqueous environment. So I briefly mentioned detergents before when we were looking at those hydrocarbon. So I had mentioned detergents before when I was talking about like how the phospholipid membrane comes together, and that if we had one hydrocarbon tail, that we would be able to create a spear. And so our detergents have one hydrocarbon tail. So by themselves, what they would do is create the spears or a detergent meniscus. The meniscus is that spear that would actually create. So when we wash our hands or we introduce a detergent to a cell, what's going to happen is the detergent is going to incorporate around transmembrane proteins and again around the lipids, and so they create these lipid detergent meniscus, right? And so this is going to break apart the cell. Okay, and if we break apart the cell can't survive, right? So if we're thinking about we have bacteria on our hands or a virus on our hands, the detergent, when we apply that to our hand, is in these meniscus. As we're rubbing our hands together, it starts to incorporate around proteins and around those lipids and create the meniscus that incorporates that. Okay, and because now we've destroyed the plasma membrane or the lipid layer of a virus, those cells aren't able to survive. Okay. So when we look at proteins that are on membranes, they can also be used to create protein gradients. This becomes really important when we're talking about the electron transport system or electron transport chain. So electron transport system is the term that's used for prokaryotic cells because this process happens on the cell membrane, on the plasma membrane. With eukaryotic cells, they have their mitochondria, and so this process is going to happen on the membranes of the mitochondria. But we can have these transmembrane proteins, and by the use of side chains, we can actually take the hydrogen and basically are passing it through this molecule so that we end up pushing protons, hydrogen ions outside the cell. And if you remember back to like an intro bio class where you talk about cellular respiration, we need to get these hydrogen ions out. We need to get these protons out so that they can come back through ATP synthase, which will cause ADP to become ATP. And again, for eukaryotic cells, this happens in the mitochondria, I would say most. For prokaryotic cells, this is going to happen on the surface. There are eukaryotic cells that do not have mitochondria. Microbiology, it's awesome. So we need to have the cell membranes to be strong, right? So that we have this definition between the outside and the inside, and that biochemical reactions that need to happen inside the cell stay there, right? But we also need them to be flexible also, right? So, if we think back to chapter 4, when there was the video about sickle cell, those cells are not flexible, right? And so we need, just the simple act of moving or exercising, standing up, sitting down, anything you do, imagine if those cells did not have the flexibility of their plasma membrane. We would all be statues, right? Because even the simple movement of your hand, as you're flexing your bicep, those muscles, as they're contracting, they're all cells, right? So those cells have to be able to respond to that change, that contraction of the muscle fibers. And if they couldn't do that, then those fibers wouldn't be able to contract, right? And your skin needs to be able, you've got all that connective tissue, but there's going to be pressures being exerted on the cells to change their shape. Maybe not change the shapes that like you physically can see that, but if we were to look at that, under a microscope, we would be able to see that. And so what we would end up seeing is that we would have proteins that would be attached to the membrane, but also attached to the cytoskeleton, right? And so that's what we're seeing in this figure, is that we have these transmembrane proteins and attachment proteins that are going to have attachments to the cytoskeleton, and we can see that in panel A and also in panel B. So panel B is microscopy. You can see that attachment, and then we can see these fibers that are running across. And so that as those filaments might lengthen or shorten, that is going to change that shape of the plasma membrane, right? So I'm trying to think of a good analogy here, right? So it's kind of like, if you have a sweater and you get a pull in it, so one of the threads gets pulled, it's going to distort and change the shape in that area. Okay, so, if you got cheesecloth laying at home, take the cheesecloth, take one of the strands of the cheesecloth and you pull it, and there's like a ripple effect. There's a whole response by that whole fabric, that cheesecloth, right? So if we think about the plasma membrane as almost a fabric that is covering the cell, if we pull in one area by the cytoskeleton that is anchored, it's going to cause changes in other areas. Okay. Related to kind of the idea of flexibility and fluidity, cell membrane proteins and phospholipids are constantly moving around inside the cell. And so a study that was done took rhodamine labeled membrane proteins in a mouse cell, and then we also had fressin labeled membrane proteins in a human cell, and they fuse the cell together, and so you create this hybrid cell. And so initially after the fusion, you can see a clear separation between the two. So these membrane proteins aren't mixed. You basically have a mouse hemisphere and a human hemisphere, but after incubation, after about 40 minutes, if you look at these cells again, these membrane proteins have moved to different locations, okay? And so there's this movement and diffusion of the proteins. In some cases, they can still aggregate together because maybe they have common, like a cell signaling pathway. So they're going to stay in those lipid rafts. But again, there's no clear distinction between the different hemispheres. And again, it shows that fluidity of the plasma membrane, how molecules and proteins and phospholipids move around within the plasma membrane, that they don't stay in one spot their whole life. So again, we want to keep in mind that some proteins are associated with parts of the membrane, and then they actually do get anchored in place. I want to point out panel C. So with cell-cell communication, we can have receptors and proteins that are going to interact with each other. So they're going to be congregated in an area, so those interactions can happen. So if we have cells in a tissue, and those tissues, these can be these connective features that are made up in tissues. And we'll talk more about these in chapter 20, I believe, when we talk about tissues and cell communication. I also want to talk about panel D. So panel D, we have more or less what is probably an epithelial cell. And so we can see at the top, the apex, we're going to have certain proteins present, because this is what's going to interact with like the lumen that's making up this area. So maybe this is like a intestinal cell, and so molecules that are important for transport and absorption would be found on that apical side, that top layer, where around the rest of the cell, the lateral and basement membrane, this is where we might have connective proteins that are going to help anchor them to the neighboring proteins. And we also see tight junctions here. So looking more closely at panel D, we have our figure here, so that we want to keep in mind that cells have their function. We have specialized cells in our body, and so depending upon what their function is, they may have different distributions of proteins. And so again, this would be an epithelial cell, so making up a tissue, and so we're going to have these tight junctions that are going to anchor these cells to each other. But then we can also have proteins or anchoring points along the lateral and basement side of the cell. On the apical side of the cell, the top of the cell, we're not anchoring to anything. This, on the top of the apical side, would be the lumen of the space. And so again, these proteins would maybe aid in the absorption of material moving through the lumen, or that are in the lumen. Okay. So again, depending upon the cell type, distributions of proteins may be different, and also expression of the proteins may be different. So that concludes the slides for chapter 11. So, for exam 1, there was more slides for chapter 4. I also kind of I gave like 2 class sessions in a way to chapter 4, where chapter 11 only has 1 class session. So I do encourage you that if you do have any questions related to this information, send me an email before our next class, so that I can make sure that we address that before your exam. Okay. There is the review that's posted up on our course page, and everybody's assigned a question. So obviously you're responsible in answering your question, but you should be able to answer all of those questions. So that is a great way to study for the exam. So also go to that, and if there's any of those chapter 11 questions that you're like, I don't understand, please make sure that you send me a message so that we can talk about that in our next class. So thank you very much.